session 5. Today, we are going to discuss chemoosmotic hypothesis or chemosmosis. And this will be discussing to understand the synthesis of ATP during cyclic and non-cyclic pathways of light phase. And we have seen, yesterday in our previous class, we have discussed about this non-cyclic pathway. Just once again, look at this non-cyclic photophosphorylation, in which two pigment systems are involved. Those are pigment system 2 and pigment system 1. And this pigment system 2 releases electrons by receiving photons, and this released electrons, they passes through various electron transporters or various electron acceptors, and finally they reaches pigment system 1. And while traveling through these electron transporters, at the point where means at the point of Cytochrome Complex, this electrons releases energy, and with the support of that energy, ATP synthesis takes place here. That was discussed yesterday in our previous class while explaining this non-cyclic photophosphorylation. These changes are discussed. So here, actually, this ATP synthesis is picturized in a short way. Actually, it is in a broad way that which cannot be shown here in this cycle. That's why we are discussing it in a separate pattern, that is chemosmosis. And in this chemosmosis, that means ATP synthesis takes place during the light reaction, and light reaction takes place in our site of light reaction is thylakoids. So if we observe the structure of thylakoid, these thylakoids are plate-like structures, and these plate-like structures are bounded by two membranes. Those are number 1, these are outer membrane and inner membrane. Look at this. This is a thylakoid which is bounded by two membranes. One is outer membrane, the other is inner membrane. Inside the inner membrane, we are observing empty place, and which is called as lumen. So this is the structure of a thylakoid. And in this thylakoid, we have to know two things. Already we have discussed about these points, and even though once again, two important things are there. One, on the inner membrane, on the inner thylakoid membrane, there is a complex. There is a complex which is called as Cytochrome Complex. And this Cytochrome Complex that helps in the photolysis of water. That means complex helps in photolysis of water. Then next, on the outer membrane, outside, there is an enzyme which is called as NADP reductase. So this enzyme is situated on the outside of the outer membrane, that means towards the stroma. Here, this thylakoid is embedded in the stroma or matrix. That's why here this point should be understood. That means NADP reductase enzyme is situated on the outside of the outer membrane. These two are the important things. First one is cytochrome complex that is located on the inner side of the inner membrane. That means in the lumen side, towards the lumen side. And another is NADP reductase enzyme is located on the outside of the outer membrane. That means towards the stroma. These two are the important things. Now, let us recall the non-cyclic phosphorylation. Look at this cycle. Once again, we should recall this non-cyclic photophosphorylation, which is a part of light reaction which takes place in thylakoids. Right. This non-cyclic photophosphorylation involves two systems. One is pigment system 2, the other one is pigment system 1, and starts with the pigment system 2. And by receiving photons, this reaction center and this accessory pigments, they absorbs the photons, and these accessory pigments, they transfers these photons to the reaction center. And in the excited state, this reaction center releases electrons which were absorbed by P6, which were absorbed by primary electron acceptor. Once again, pigment system 2 involves reaction center. Pigment system 2 involves in pigment system 2 this chlorophyll pigment, which is treated as a reaction center, absorbs 680 nanometers of wavelength, red color wavelength, and this accessory pigments also receives or also absorbs the photons of 680 nanometers of red color wavelength or red color light. So by absorbing this photons, reaction center chlorophyll gets excited and releases electrons. These electrons were accepted by primary electron center. And electrons are transferred through plastoquinone, cytochrome complex, and plastocyanin, and finally it is accepted by PS1 chlorophyll A or PS1 reaction center. This is the normal process. And here 3 electron acceptors are there. Electron transporters are there. They are plastoquinone, B complex, and plastocyanin. And here electrons were passed through plastoquinone, cytochrome complex, and plastocyanin. And here taking up this cytochrome complex only, where exactly that means where exactly this ATP synthesis takes place. Electrons are transferred from plastoquinone to cytochrome complex and the plastocyanin, and from there it is accepted by reaction center 1 of PS1. But this phosphorylation is situated or is located at this complex area only in non-cyclic photophosphorylation. So when these electrons uh, they travels or they are transferred from higher level to lower level, uh, lower level, they releases energy. That means electrons they reaches to primary electron acceptor with high energy gradient, and they passes through various electron acceptors and finally reaches to PS1. But the here electrons moves with a high energy gradient, and they move towards a low energy gradient. So in this travel at the point of this cytochrome, at this point, while passing electrons, to PC, that means receiving from plastoquinone and passing it to PC. So while passing the electrons to PC, this electrons releases some energy there, where means at a cytochrome complexer area. So here, with this energy, now come to the chemosmosis cycle here. With this energy, cytochrome that drags or pulls protons into the lumen. This cytochrome complex, this by utilizing the energy, it pulls the protons from stroma into the lumen. By using this energy only, it is possible. That means here the energy is released by the pair of electrons. Energy which is released by pair of electrons. Energy which is released by pair of electrons. When these electrons are coming down to the gradient, this energy is needed used by cytochrome. Here once again, electrons which are released by reaction center of PS2 are accepted by primary electron acceptor, and from here with a high energy gradient, this electrons moves to the low energy gradient, and while traveling or this journey is through the electron acceptors, finally reaches the PS1. But when the electrons moves from plastoquinone to cytochrome complex, there is no change. But here at the point of cytochrome complex, this electrons releases nothing. But when the electrons moves towards the plastocyanin, this electrons releases some energy, and this energy, with the support of this energy complex, that pulls the protons, from stroma into the lumen of the chloroplast. That is first point. That means here what we have to understand is, in chemosmosis mainly two steps are involved. First one is increasing the proton gradient in lumen, and how it is or how it will be means in this way only. During the non-cyclic photophosphorylation, electrons are passed through various electron acceptors and finally reaches PS1 reaction center. And while passing through these electron acceptors, at the site of cytochrome complex, by releasing energy, electrons are moved towards plastocyanin. And by using this energy, cytochrome complex drags the protons or pulls the protons into the lumen of the thylakoid. So in one way, these protons are and protons are entered into the lumen. Right. Here it is indicated by in this way. See here, pigment system 2 and pigment system 1. Both are involved in non-cyclic photophosphorylation, and this non-cyclic photophosphorylation takes in membranes of thylakoid or in thylakoids. So these are located in the membranes. Now, for pigment system 2 starts by accepting the protons from the sunlight, and also in addition to that, accessory pigments, they transfers the photons to the pigment system 2. So by receiving these photons in excess, reaction center chlorophyll A gets excited and releases electrons, and those electrons are transferred, those electrons are accepted by PEA, primary electron acceptor. And from there, from high energy gradient, this electrons moves downwards through various electron acceptors, and finally those are accepted by photosystem 1 reaction center. So here, while transferring from high gradient energy to low gradient energy, these electrons, when they are moving from different electron acceptors, the right side of this ATP synthesis or protons absorption is located in the center of cytochrome complex. Why means, while passing the electrons from cytochrome complex to plastocyanin, this electrons releases some energy here. So by utilizing that energy, this cytochrome that drags the protons from stroma into the lumen. So that is one step. In the other step, here in photosystem 2, there is an electron loss. Why means, whatever electrons are released during the excited state are transferred through various electron acceptors, and they are accepted by photosystem 1, but not by the photosystem 2. That means automatically there is there will be electron loss created. So this situation creates electron loss in the photosystem 2 reaction center. So to continue this photosystem 2, there is a need of fulfilling electrons. Then how it will be? That means this to replace the electron pair in photosystem 2 is possible only by photolysis of water, and that is possible by the cytochrome complex, which is situated on the inner surface of the thylakoid. So with the support of this thylakoid complex, H2O splits into here. Yes, here H2O splits into H plus and OH minus, and OH minus furtherly splits into oxygen and the electrons. Here, 3 end products are produced. They are pair of electrons and pair of protons and half molecule of oxygen. Out of these 3, oxygen that diffuses out as a byproduct, and protons, whatever electrons, what about the electrons? These electrons are accepted by pigment system 2 reaction center. That means electron loss is fulfilled by the photolysis of water electrons. Then next, what is the remaining end products? Means protons. Protons are in the lumen. They are produced in the lumen. That means on the whole, photolysis of water is taking place in the lumen only. That's why protons are produced in the lumen. So in one way, protons are pulled by the cytochromes into the lumen. Then in another way, by photolysis of water, also the protons are produced in the lumen. That means here proton concentration increases. That means how the proton concentration is increasing in lumen. In lumen, proton concentration is increasing in two ways. One is pair of protons which are produced by photolysis of water. Then next, this cytochrome complexer. Cytochrome complexer that pulled or that pumped the protons from stroma into the lumen. So in both the ways, protons are gathered in this lumen. By that, this results in the accumulation of protons in the lumen. So in two ways, this proton concentration is increasing in lumen. One way from the stroma by the support of cytochrome, they are pumped into the lumen. In another way, photolysis of water is also taking place on the inner membrane of the thylakoid, which results in the formation of three end products. Out of those three, one is oxygen, which diffuses out as a byproduct, and the electrons, second end product electrons, which compensates the electron loss in the pigment system too. And the remaining is protons, pair of protons. So automatically they are in the lumen only. That means this protons, photolysis of water resulted in the formation of protons are in the lumen only. So in this way, in two ways, protons are accumulated in the lumen. So protons concentration that increases in the lumen. As a result of that, reduction in the level of proton concentration reduces in the stroma. That means when the protons are pumped into the lumen, automatically there is a reduction in protons concentration. So this results concentration gradient. That means proton concentration gradient. That means a high concentration in lumen and a low concentration in stroma. So this is the situation right now. Then what happens? This situation leads to what? That is in the next step. Up to here, once again, repeating, that is during non-cyclic photophosphorylation. During non-cyclic photophosphorylation, a reaction center releases electrons by absorbing photons. Not only by reaction center, this accessory pigments also helps in receiving photons and transferring them to reaction center. As a result of this absorption of photons having 680 nanometers of wavelength, red light, when it is absorbed by reaction center, it gets excited and releases electrons. These released electrons are absorbed by primary electron acceptor. And from here, with a high energy gradient, this electrons moves through electrons are transferred through various electron acceptors or electron transporters, and finally these electrons are accepted by a reaction center 1. And while traveling from high gradient energy to low gradient energy through these electron transporters, there is a site which is important, that is cytochrome complex. There, when the electrons comes from plastoquinone and reaches cytochrome complex, and from there, this electrons, by releasing some energy, they moves towards the plastocyanin, and this released energy helps, or this released energy helps in dragging the protons from stroma into the lumen. That is an important issue which is explained in detail in chemoosmosis. Here, this in non-cyclic, two systems are involved: photosystem 2 and photosystem 1. Started with the 2 only, and this system 2 reaction center releases electrons, and this electrons moves through various electron acceptors and finally reaches system 1. And while traveling through this electron acceptors, here one important issue is possible at cytochrome site, and this cytochrome complex at this point, this cytochrome complex receives some energy by the electrons when they leaves this cytochrome complex and moves towards plastocyanin. And this released energy by the electrons is utilized by the cytochrome complex in dragging the protons from stroma into the lumen. So that is an important issue during the non-cyclic photophosphorylation. And this was not possible in sketching there in the cycle. That's why we are discussing it in separate diagram. So here, in one way, the protons are entered into the lumen. In another way, there is another way which helps in increasing the protons concentration in lumen. That is nothing but photolysis of water. And this photolysis of water, for this photolysis of water, one complex is helpful, that is cytochrome complex, which is situated on the inner side of the inner wall. And this complex helps in splitting water molecule into H plus and OH minus, and OH minus further splits into oxygen and the electrons. And this protons remains in the lumen only, and oxygen that will be diffused out as a byproduct, and electrons are received by the photosystem 2 reaction center where electron loss was there. So to compensate that, the electrons are useful. And as a result of these two events, protons concentration or accumulation of protons in the lumen takes place. That means protons concentration increases in the lumen than the stroma. That means a reduction in the level of proton concentration in stroma, whereas increase in protons concentration in lumen. Both are possible by these two activities. One is by utilizing the energy which was released by electrons while passing to PC, helps this cytochrome complex to drag the protons into the lumen. That is the one way of protons entry into the lumen. On the other hand, photolysis of water also helps in producing the protons in the lumen. These both situations helps in increasing the proton concentration in lumen. This is very clear. High concentration in the lumen and low concentration in the stroma. So in this situation, proton shows interest in coming out into the stroma again. That means concentration gradient is situated between lumen and stroma. So from high concentration to the low concentration, this proton molecules shows interest in coming out into the stroma. That is possible only when there is protein particles. That will be discussed in further steps. Here, the situation, what type of situation is created means from this high concentration lumen, proton high concentration lumen loses the protons into the low concentration stroma. That means in lumen, protons concentration is more, whereas in stroma, protons concentration is less. That's why this proton shows interest to make a move into the stroma, from lumen into the stroma. And for this transfer, it requires, that means for this transfer, it requires some protein particles. And these protein particles, they acts as channels for transferring these protons into the stroma. So for proton transfer from lumen into the stroma, it requires two different types of protein particles. And those are, see here. Those are number one, the F0 particles. Here, F0 particle, which are called as integrated embedded protein particles. This F0 particles, they are embedded in the two membranes of thylakoid. It is shown in the diagram. See, F0, it is in between the outer and inner membranes of the thylakoid. That's why it is called as embedded, integral embedded protein particle, which is called as F0. And the other one is here, it is in the form of a protrude, or it is appearing like an outgrowth from the F0. See, which is situated in the stroma, not in thylakoid. F0 is in between the lamellas of thylakoid, but the F1 protein particle, it is appearing like an outgrowth from the F0, and it is in the stroma, situated in the stroma. And this F1, it acts as an enzyme, which is called as ATPase. ATP synthetase. And this ATP synthetase enzyme helps in synthesizing ATP by adding ADP with IP and to result in the formation of ATP. So F1 particle, protein particle, is acting as an enzyme, and that enzyme name is ATP synthetase. Okay, these two are the important protein particles for transfer of protons from lumen into the stroma. That means here, these protein particles, they are acting as channels. They are acting as channels, or they are acting as passing ways for this protons, or for the movement of protons from high concentration lumen into the low concentration stroma. This is very clear. Here, by the photolysis of water and by cytochrome complex, by utilizing the energy by cytochrome complex, results in dragging or pumping the protons in from stroma into the lumen. And these two activities helps in the accumulation of protons in the lumen. So there is a proton gradient situation is created in the lumen. That means proton concentration is high in lumen and low in stroma. And at this juncture, this protons shows interest in coming out into the stroma. And this is possible only through protein particles. There are two types of protein particles, which helps in transferring stroma. So these protons, they makes movement through special protein particles, which are called as F0 and F1. F0 protein particles are arranged within the or in between these two membranes of thylakoids. And F1 particle, it protruded out into the stroma, or it is appearing like an outgrowth. It is looking like an outgrowth in the stroma. And this F1 particle acts as an enzyme which is called as ATP synthetase or ATP synthase. And this helps in ATP synthesis. Now, this situation was created by the protons, and this protons now makes a movement through this F0. How it is? When the proton pair moves, when the proton pair moves from lumen through the F0. Here, these protons, they move towards the F0 and they passes through the F0. And for passing through this F0, they do not spare any energy. That means F0, it helps in facilitated diffusion of protons. That means facilitated diffusion means here facilitated diffusion means diffusion of any substance across a membrane depends upon its solubility in lipids, which also move faster. And substances which have a hydrophilic moiety find it difficult to move through the membrane. Their movement has to be facilitated by membrane proteins without utilizing metabolic energy, and there must be a concentration gradient. That is called as facilitated diffusion. By all means, what we can understand means here for the transfer of this protons, pair of protons through this F0, and this F0 helps in creating facilitated diffusion. Why? Because concentration gradient situation is there. They are coming from high concentration. So with high energy, this electrons comes into the F0 channel, and there this F0 protein particle creates a facilitated diffusion. That means without sparing energy or without investing any energy, these protons, they move from a lumen through the F0. That means there is no energy, there is no energy requirement, or energy is not required for the diffusion of protons from lumen through the F0. Right. Now, after crossing this F0 protein particle, protons, they enters into the F1 protein particle. So protons moves with energy, and that energy is here in F1. Protons moves with some energy, and this energy, that means here F1, this protons, they comes from F0 into F1. While moving from F0 to F1, these protons, they releases some energy, which is utilized by F1 ATP synthase to combine one ADP with one IP to synthesize one ATP molecule. That means here one ATP molecule is synthesized, and this electrons with little energy, they enters into the stroma. Okay, that means once again from high concentration gradient, this protons makes a movement through this F0, which is embedded in the membranes of the thylakoid. And here this F1 facilitated diffusion. That means there is no energy is required for transferring of these protons from lumen through the F0 into F1. So energy is not used while passing through this F0. And with that energy, this protons moves through the F1 into stroma. And during that moment, or at that moment, this protons releases some energy. That energy is utilized by F1 ATP synthase enzyme in adding one ADP with IP in synthesizing one ATP molecule. That means phosphorylation, actual phosphorylation takes place at this juncture. In non-cyclic, what we have discussed in our previous class is ATP synthesis takes place at the site of cytochrome complex. That is shown in a simpler manner. But actually, how the ATP synthesis takes place means in this complicated way only ATP synthesis takes place. That means while electrons while transferring from one electron acceptor to the another acceptor, they passes through the cytochrome complex. But passing from cytochrome complex to PC, this electrons releases energy. With the support of that energy, cytochrome complex pumps the protons from stroma into the lumen. That is their actual process is that. And after pulling these protons into the lumen, and by the result of photolysis of water, this protons concentration increases in lumen. That means the concentration gradient is created in this lumen. That means high concentration of protons in the lumen, low concentration of protons in the stroma. In this situation, protons, they shows interest to make a move into the stroma again. So this movement is possible only when there are this special type of protein particles, which are called as F0 and F1. So when the this F0 is called as embedded protein particle, which is called as embedded. Why it is called as embedded? Because this protein particle is arranged in between the two membranes of the thylakoid. That's why it is called as embedded. Then next, F1 is called as a protrude. That means it is looking like an outgrowth from the F0. So both these protein particles, they are acting as channels for the transfer of protons from lumen into the stroma. And here F1 especially, it acts as an enzyme synthetase, ATP synthase. And this helps in adding ADP with one IP in the formation of ATP. That means actual phosphorylation is taking place in stroma only. Then next, one more point here is there's electrons which are accepted by pigment system 1, and they too receives the photons from the sunlight and undergoes the excited state. And under excited state, this reaction center again, it releases electrons, pair of electrons, which are accepted by primary electron acceptor. From there, it passes the electrons to FD, and from FD, these electrons are passed to NADP plus to reduce it. Here, this NADP plus should be reduced. For this, it requires protons, and from where they will receive the protons. That means here protons are coming. These protons are coming from lumen into the stroma. So there is a flow of protons from lumen to the stroma, and these protons are Here, this protons. So here through this protons, they are connected with the NADP plus to reduce it into NADPH2. So in this way, while discussing this non-cyclic, what we have discussed is assimilatory power is produced or synthesized during non-cyclic photophosphorylation. That means what is assimilatory power? ATP plus NADPH2. So here we are observing the synthesis of ATP in the stroma only and NADPH2 synthesis also in the stroma only. So these are togetherly called as assimilatory power. And this assimilatory power or ATP and NADPH2, they are where are the ATP and NADPH used? That means utilization of this assimilatory power where it is required means this assimilatory power is required for this dark phase or Kelvin cycle, which takes place in stroma only. So assimilatory power is synthesized in stroma. See here, assimilatory power, ATP and NADPH2, both are synthesized in stroma only. And in stroma part, that phase takes place. The other phase of photosynthesis, which is called as dark phase, and it was invented by Melvin Kelvin, that takes place in the stroma. So for that, Melvin Kelvin's sake or Melvin Kelvin cycle absorbs the assimilatory power from stroma. By this, we can understand that dark phase takes place in stroma part of a thylakoid. So here, one important thing that is this chemosmosis that takes place in chloroplast as well as in mitochondria also. And in both in mitochondria and chloroplast, this complex is associated in photolysis of water. That means in chloroplast, it is having cytochrome complex on the inner surface of the inner membrane. And in mitochondria also ATP synthesis takes place. And that is associated with cytochrome complex only. So this chloroplast and mitochondria both are helpful in producing ATP, which are associated with cytochrome complex. That is an important point. So here, why the chemosmosis? That should be understood by the students. So dear students, try to understand what is chemosmosis. Chemosmosis is the main concept of this chemosmosis is where and how the assimilatory power is synthesized actually. That means is the cytochrome site for the synthesis of ATP or is a part of this process. That is very clearly explained in chemosmosis. So once again, during this non-cyclic photophosphorylation, whatever electrons are transported through electron carriers and finally reaching by accepting by reaction center 1. During this journey, electrons, they moves from cytochrome complex to plastocyanide, and at this moment they releases some energy, and this energy is utilized in pumping the protons from stroma into the lumen, and that is shown in this diagram. Right here. So from PS2, which absorbs 680 nanometers of wavelength, red light is absorbed by pigment system 2, and in the form of photons, they receive the light. And by undergoing excitement, this system releases electrons. They travel through various electron carriers or transporters, and finally they reaches the pigment system 1. They are absorbed by pigment system 1. So during this journey, electrons from leaving this cytochrome complex and reaching the plastocyanin, they releases some energy, and this released energy helps in dragging the pulling the protons from stroma into the lumen. In this way, protons are entered into the lumen. In another way, that is nothing but photolysis of water, and this is associated with the cytochrome complex, which is situated on the inner surface of the inner membrane. Splits the water molecule into 3 end products: pair of electrons, pair of protons, and half molecule of oxygen. Oxygen diffuses out as a byproduct, and electrons, they replaces the electron loss in pigment system 2, and protons, they remains in the lumen only. As a result of these two actions, that means by utilizing the energy, cytochrome complex dragged pumped the protons into the lumen, and photolysis of water also released the protons into the lumen. So by this, concentration of protons increases in the lumen than the stroma. So there is accumulation of protons, which creates a high concentration of protons in the lumen and a reduced concentration of protons in the stroma. And at this moment, this lumen protons, they shows interest in passing through the, they shows interest in entering into the stroma through some particular protein particles, which are called as F0 and F1. F0 is called as embedded protein, and F1 is called as ATP synthase. And this F0 protein particle is embedded in between the membranes of the thylakoid, and it helps in transferring the protons from lumen to the F1, and during while passing these protons, it helps in facilitated diffusion. That means no energy is required for passing these protons from lumen into the F1. And this F1, after passing through this F0, this protons with energy, they moves through this F1, and while passing through this F1, they releases some energy, and this energy is utilized by ATP synthase enzyme in adding one ADP with one IP in the synthesis of one ATP molecule. So here phosphorylation is taking place in stroma, and this released protons, they helps in reducing this NADP plus. And before this, we have to see the pigment system 1. It receives the electrons from pigment system 2, and also it receives the photons from sunlight, and also with this accessory pigments also it receives the photons. So by all this, it receives more than enough photons. So because of that reason, it undergoes excited state, and under that excited state, P700 releases electrons, and this pair of electrons are received by again primary electron acceptor. From there, with high energy gradient, this electrons moves towards the pyridoxine, and this pyridoxine again gives the electrons to the NADP plus to reduce it into NADPH2. So here ATP and NADPH2 both are synthesized, and these two are togetherly called as assimilatory power. And this assimilatory power, where it will be utilized, that means in stroma, we know it. We know this fact. What is that? Kelvin cycle or dark phase of photosynthesis takes place in stroma only. So to perform this stroma, light is not required, but energy is required. That energy is assimilatory power, and this assimilatory power is utilized by this Kelvin cycle. That's it. Okay, bye. Okay, students, we'll stop it here, and tomorrow we'll continue the other. Thank you.